Hello everyone. This is the GIS Biochemistry Topic 2 review, which is about the digestion and absorption of lipids. It's Bashar al-Marzik from the Hashemite University, and now let's get in. As always, this video is a summary of Topic 2. Use it for revision purposes only. Every single detail is mentioned in the main lecture, so go back whenever you need. Here we go now. First, with a short intro to lipids. Lipids have some characteristics that make them unique from other macromolecules. They are relatively insoluble in water. They are soluble in non-polar solvents. We refer to them as relatively insoluble because short-chain fatty acids are soluble in water while long-chain fatty acids are not. Compared to carbs, lipids store much more energy because they are rich in hydrogen-carbon bonds. Functionally, lipids are a storage form of energy. They are components of cell membranes and act as electrical and temperature insulators. Furthermore, they help in the absorption of fat-soluble vitamins, which are A, D, E, and K. Lipids, when eaten, will travel along the alimentary tract and will get along the following steps, where the first 3 steps are digestion and the last 2 steps are absorption. Let's have a look at the digestion of these hydrophobic molecules. Lipids have to be hydrolyzed and emulsified into very small droplets called micelles before they can be absorbed. And first, they will be processed in the stomach. Hydrolysis of triacylglycerols is initiated by the lingual and gastric lipases, which attack the third ester bond forming 1,2-diacylglycerols and free fatty acids, aiding emulsification. They are specific for short and medium chain fatty acids and will work optimally at pH of 3 to 6. These 2 enzymes are important in neonates since the pH in the neonatal stomach is higher than that of adults and milk is rich in short and medium chain fatty acids. Second, the emulsification process takes place. It is defined as the breakdown of fat globules in the duodenum into tiny droplets, which increases the area of contact between lipase enzyme and lipid droplets, thus increasing the rate of hydrolysis. It occurs due to the peristalsis and it's helped by presence of bile salts as shown in the following figure. The last step of digestion involves 3 enzymes, the pancreatic lipase, intestinal lipase, and an isomerase. These enzymes act by attacking the first and third bonds for pancreatic lipase, which is in need for the protein colipase to function, the first bond in monoacylglycerol for intestinal lipase, and lastly, isomerase enzyme helps by moving the acyl group from the second position, which is indigestible by the above enzymes, into the first position on triacylglycerol. By the end of this step, we will have 2 monoacylglycerol, glycerol, 1 monoacylglycerol, and free fatty acids. The following table have all the important information summarized. The last 3 enzymes are for the digestion of phospholipids, lysophospholipids, and cholesterol esters respectively. The end products are lysophospholipids, glycerophospho-X, and cholesterol with free fatty acids respectively. For the regulation, we have cholecytokinin, CCK, which is secreted by duodenum to reduce gastric motility, upregulate pancreatic enzymes, and induce gallbladder contraction to release bile. On the other hand, we have secretin, which is produced in response to acidic chyme to release watery solutions rich in bicarbonate to neutralize pH. After having the lipids ready to be absorbed, we start the uptake process and resynthesis. The mixture of lipids absorbed by the enterocytes migrates to the smooth endoplasmic reticulum where biosynthesis of complex lipid takes place. Long-chain fatty acid will be activated first by thiokinase, acyl-CoA synthetase, forming acyl-CoA, which is used for the reesterification of the absorbed 2 monoacylglycerols, forming triacylglycerols. The 2 MAG absorbed by the enterocytes are converted to TAG through sequential reacylation by 2 acyltransferases: acyl-CoA mag acyltransferase and acyl-CoA diacylglycerol acyltransferase. Cholesterol is united with active acyl-CoA by the aid of acyl-CoA cholesterol acyltransferase ACAT to form cholesterol ester. Short and medium chain fatty acids are not converted to their CoA derivatives and are not re-esterified to 2-MAG. Instead, they are released into the portal circulation, and the figure represents actions on long chain fatty acids. Lastly, long chain fatty acids are esterified to yield to triacylglycerol in the mucosal cells, and together with other products of lipid digestion highlighted in the slide, secreted as chylomicrons into the lymphatics, entering the bloodstream via the thoracic duct. Short and medium chain fatty acids are mainly absorbed into the hepatic portal vein as free fatty acids. For defects in the mentioned 5 steps, all end as steatorrhea, which is lipids in the stool. These defects are deficiency of pancreatic lipase, which occurs in chronic pancreatitis, cystic fibrosis, obstruction of the pancreatic duct by tumors, and after pancreectomy, in addition to severe hyperacidity. Lipids in the stool are being undigested in this defect. Deficiency of bile salts, which occurs in tumors or stones of the bile duct, hepatitis, and liver cirrhosis. The lipid of the stool is digested. Defect in intestinal epithelium, including malabsorption syndromes, with the lipid in stools being digested. And lastly, A-beta lipoproteinemia, in which ApoB48 cannot be incorporated into chylomicrons. This caused the accumulation of triacylglycerols. Finally, absorbed lipids will have the following fates: lipolysis of triacylglycerol, which will produce glycerol and free fatty acid. Glycerol will form triacylglycerol and will enter gluconeogenesis via the molecule dihydroacetone phosphate. Fatty acid will be used by the muscles for energy purposes and also will be taken up in the adipose tissue. Remnant chylomicrons will be endocytosed by the liver.